So in this next recording on molecular and cellular biology, I want to dive a little bit deeper into proteins and spend a little time talking about protein folding. Again, proteins are one of our four main classes of biomacromolecules: carbohydrates, lipids, proteins, and nucleic acids. And we know that proteins are made of amino acids, that we can link amino acids together with a peptide bond, and that ultimately that's the primary structure, which then folds into microstructure or secondary structure, which then packs into tertiary structure, the three-dimensional shape of the protein. And this introduces the idea that we're going to see in biology again and again, is that form is function. And so let's look at an example of sickle cell disease, where when we change the primary structure of a protein, we change the shape of the protein, and hence the change of the shape of the protein changes its ability to do its job. So sickle cell disease is a genetic disorder. It's a point mutation that results in amino acid substitution. So here what we can take a look at is the beta subunit of hemoglobin. So hemoglobin as a protein would be considered a protein with quaternary structure. It's made of 4 polypeptides, 2 alpha subunits and 2 beta subunits. And at the top, this primary structure is listed: valine, histidine, leucine, threonine, proline, glutamine, glutamine. Right? So what's going to happen in the case of sickle cell is that we're going to change that 6th amino acid to a valine. And what you can see is that changes, that's in a key location. And because of that key location, it's going to change the shape of that beta subunit. Now those beta subunits, when they engage in the protein complex that is now sickle cell hemoglobin, they tend to glom together and create these long fibers inside the cell. They don't carry oxygen nearly as well, and the resulting phenotype of the cell, or the physical type of the cell, is these sickle-shaped cells. So you can see here on the left a normal red blood cell compared to the sickle cell red blood cell. So we can see clearly here a single change in the primary structure can, in some cases, have a drastic effect. Right? So what determines protein structure? Laws of physics, right? So physical and chemical conditions can affect protein structure. Proteins have evolved to maintain their proper shape in the environment that they're going to do their job, right? So alterations in pH, salt concentration, temperature, the concentration of the protein itself or other proteins around it, or other environmental factors can all lead the protein to lose its shape. And the special word we use in protein chemistry for that is denaturation. So a protein is denatured, has lost its native or normal structure, and now is a dead protein. Okay? So form equals function. So we can depict that a couple different ways. When we have this ribbon display of the protein, you can see a normal protein here on the left, and then when it's denatured, those ribbons become unfolded. It is a denatured protein. It has lost its shape. And because it's lost its shape, it can't do its job anymore. So some proteins we can renature, they can fold back into their proper form, but not always. So let's look at that form and function in a little bit more detail. And so this is a particular protein called lysozyme, and you can see this groove is labeled here. And we'll talk about this more as we talk about enzymes later in the quarter, but that groove is going to be what's called the active site of the protein, and it has a very, very specific shape. And so you can see that in the ribbon model or the space-filling model. We can zoom in on that. It's a very specific shape. Again, it's the shape of that groove that allows it to bind to its substrate to do the chemistry it needs to do as a catalyst, as an enzyme. Right? So what we always want to remember, it's the sequence of amino acids that determines a protein's three-dimensional shape. And it's that shape that dictates its function. And this can be incredibly specific. So if we look at antibodies, which are proteins, and how they bind to invaders, in this case a protein from a flu virus, it's the shape of that interface where the antibody binds to the flu virus protein that allows it to recognize it and bind to it and tag it and say, hey, this is an invader. Where does that shape come from? It comes from the primary structure, the amino acid sequence, and that amino acid sequence ultimately comes from your genes. So let's talk a little bit more about protein folding then, and realize that this is a complex process. So we have not yet, though people are working on this very hard, to say here is any given sequence of amino acids, what does its three-dimensional shape look like? We can't do that quite yet, and we have some very, very powerful computers working on that, but we can't do it. It's very complex. Some proteins will fold spontaneously. You can synthesize the amino acid chain, put it in an aqueous environment, boom, and it'll fold right up. Some you are so robust that you can actually heat them up, denature them, and they'll fold right back up when you bring them back to room temperature. Now, not all proteins are that robust, and some need help to fold properly. So, of course, because proteins do stuff, there's a whole nother class of proteins, a whole family of proteins that are involved in helping other proteins fold properly. And one group are called heat shock proteins. Another group are called chaperonins or chaperone proteins. So if we consider the folding pathway of a protein, we could think of it as a linear pathway. So you at the top here representing an unfolded protein, and then the I's being the intermediate forms to the native protein on the right. You could envision this as a process of very specific forms proceeding one to the next until you get to the native state. And this may be the case in some situations, but in most situations it's probably much more complex, where the number of unfolded states is probably very high, and the pathway to get to the native state is probably relatively complex, and there's usually more than one way to do it. All right? So you can measure the kinetics of this protein folding. And again, this is a very, very interesting topic that a lot of people are studying. And you can say, well, how long does it take to go from this unfolded state to these individual intermediates to ultimately the native state? And you can see this now with BPTI, which is bovine pancreatic trypsin inhibitor. It's a very useful protein in many assays, and so it's very well studied. The other thing that's very interesting about it, it has lots of disulfide bridges, and so these help to hold the protein together. And so people have tried to analyze, well, in what states intermediates does it go through before it gets to its native state? And we've seen that it goes through several different intermediates, and again, each of those arrows is double-headed. It's moving back and forth, right, based on concentration, based on the kinetics of this folding. Now, when we think about those kinetics, we can also think about energetics, right? So we'll talk about Gibbs free energy a little bit later in the quarter as well, but think of it now as a state of energy, potential energy in a molecule. And in this case, the more energy this protein molecule has, the more unhappy it is, because this unfolded state is going to expose lots and lots of hydrophobic patches to the aqueous environment, and we already know about hydrophobicity. Those hydrophobic amino acids don't want to interact with water. The second law of thermodynamics is trying to drive this to a state of greatest disorder, and that's going to be a state where those hydrophobic patches are not interacting with water. And so what we're looking for is this kind of global energy minimum. That's how we talk about it, and it's at that state where the protein now is most stable in its folded native form. And we can depict that as an energy landscape. And this is where things get very interesting and the complexity of biology really kicks in, because you have many, many unfolded states, right? And ultimately, they're moving downhill towards a lower energy state in that minimum that is folded, that F, that red part in the center. But you can also notice that some of these intermediates, these misfolded states, are little dips in and of themselves. And sometimes proteins get stuck in that misfolded state. And again, biology has evolved to compensate for this. And so, for an example, a chaperonin is a whole protein complex to help that protein get out of its misfolded state and into its correctly folded form. And so this is a large barrel-like protein that literally takes the misfolded polypeptide into its interior and literally massages that in a new environment. And that environment includes some hydrophobic patches, some hydrophilic moieties, but to try to give that protein an opportunity to get out of that well of misfolded energy and down to its global minimum, and then it can be spit out and we can do another protein. It's enzymatic, right? So proteins are very important, but their shape is ultimately what dictates their function, and that's what we want to remember. Now, you'd think that proteins and lipids and carbohydrates were complex enough, but what we can really do is start to talk about combining them. And this is much more abundant in biology, is seeing proteins that are glycosylated or lipids that are glycosylated, and that's a fancy word to say that they have sugars added to them. So a glycoprotein is a protein decorated with sugars. A glycolipid is a lipid decorated with sugars, and it allows you to diversify. It allows you to specify molecules or lipids. This is one mechanism by which your immune system recognizes self as opposed to other. And so you can see here, this is a diagram of the cell membrane where you can see proteins, and you can also see proteins attached to carbohydrates. You can see lipids, and you can see lipids attached to carbohydrates, right? So the glycolipid, the glycoprotein, right? So, and in addition, this is a whole set of biology that we call glycobiology. And you can learn about how different carbohydrates are attached at different sites to different proteins. And if that's interesting to you, I encourage you to read more deeply on this topic. So with that, then we're going to end our survey of proteins right now, and then we'll finish up our biomacromolecules with our next recording.